Before I begin the Kelvin cycle, I want to do a quick overview of the light-dependent reactions. Again, it's like part one of photosynthesis. So the important things that you need to understand about the light-dependent reactions is that obviously you need light. Light is going to hit and excite the electrons that have been removed from water. Water is ultimately split. Oxygen is a byproduct and is given off. Water being split also allows for a hydrogen ion concentration to build up inside the thylakoid space, the space that's in the inside of it, which is going to cause a decrease in pH. Okay, so that's a little bit of background that you need to know. Now, when the electrons get excited, they bounce around through the electron transport chain, and they make their way to photosystem 1, which is the second part of this. And I know the numbers are backwards. The electrons get re-excited. The goal of the light-dependent reactions is to generate the electron carrier NADPH and some ATP for the Kelvin cycle, for ultimately glucose to be made. So the purpose of the light dependent reactions, again, is for that NADPH, which is going to go to the Kelvin cycle, and ATP. Now at the bottom you can see an ATP synthase, an enzyme that directly generates ATP by oxidative phosphorylation. Because the hydrogen ion concentration is building up inside of that thylakoid space, ATP synthase is able to pump the hydrogen across the membrane, and that allows ATP, that motion of the hydrogen moving across, allows that enzyme to turn, and it adds a phosphate directly to an ADP, which forms ATP. So hopefully that's an overview. Hopefully that's something that you already know. And then we're going to go into the second part. Again, Kelvin cycle, we need to make glucose. So a summary of the Kelvin cycle. This is where ATP and NADPH are going to convert carbon dioxide to sugar. This process uses ATP, it uses NADPH and carbon dioxide. What is going to be produced isn't directly going to be glucose, a 6-carbon sugar, but rather it's going to create a 3-carbon sugar, which will be put together with another 3-carbon sugar to make a glucose molecule. The name of the 3-carbon sugar that is generated from the Kelvin cycle is G3P, glyceraldehyde 3-phosphate, which again with these crazy names, it's better just to use the abbreviation G3P. And make sure you understand that G3P is a 3-carbon sugar. There are 3 phases of the Kelvin cycle. First, carbon fixation. Second, reduction. And third, a regeneration of RuBP, which is a carbon dioxide acceptor, and I will go over that when I have the diagram. So phase 1, carbon fixation, is where carbon dioxide needs to enter into the cycle. Carbon dioxide entering the cycle, only one of them are going to enter in at a time. So depending on the diagram, you might see a diagram showing you the net or the overview, like this diagram is going to. It says up at the top, 3 carbon dioxides are going in. So when we start looking at the net ATP that's getting used, the net NADPH getting used, it's for all 3, but only 1 go in at a time. So the first thing that happens is 1 carbon dioxide joins up with a molecule RuBP. So they join up, so that way the G3P can eventually be made. Now, what causes these 2 things to react with one another, the carbon dioxide and the RuBP? An enzyme known as Rubisco. So Rubisco adds up those, or gets those 2 molecules to join up together, which is part 1 of the Kelvin cycle, again, known as carbon fixation. It's fixing carbon dioxide to that ρBp. Part 2 of the Kelvin cycle is known as reduction. So again, remember, reduction is a gain. So what is it gaining? It's gaining phosphate from ATP. So ATP is going to become ADP. The other thing that it's going to be gaining is hydrogen from NADPH. So the intermediate phase, which you don't have to know all the intermediate. No, buddy.
Please.
Okay, go. Yes, come here. Say hi.
Hello, World Heaven Hassle. My name is Will Pat.
Okay, bye. Okay, hurry up.
I'm gonna sleep all down long.
Sleep? Okay, bye. Go sleep upstairs. Okay, so you don't have to know all the crazy intermediate names of these molecules for the reduction phase, but what you do need to know is that this is where ATP is being used and the NADPH is being used. And the molecule in between is being reduced by gaining parts from those energy molecules or from those electron carriers. In the end of this phase, we have that 3-carbon sugar, which is G3P, which is released, which will then go and turn into a glucose molecule when it joins up with another one. Because the Kelvin cycle is a cycle, something has to be regenerated. So the molecule that's going to be regenerated is ρBp, again, ribulose biphosphate. So that's one of the crazy names that you do need to know. And in this process of that being regenerated, it also uses ATP for that process. So at the end of the cycle, we again have the ρBp, which will join another carbon dioxide thanks to the Rubisco enzyme, and then the cycle can occur again. Now, I think I said this on the first slide. Sometimes you're going to see this as 1 carbon dioxide going in, and it's going to say, oh, the cycle has to take place 3 times. The reason that it has to take place 3 times, 1 carbon dioxide has 1 carbon. So remember, always try to follow those carbons. So it has 1 carbon, G3P, which is going to be an important piece to glucose, has 3 carbons. So this has to take place 3 times in order to make 1 G3P molecule.